Hello, everyone. My name is Robb Krumlauf from the Stowers Institute. Let me start by apologizing for my voice. This has been such a wonderful meeting that I basically am losing my voice from all the discussions at poster sessions and stuff. And of course, I know I should save my voice, but it's so exciting, I just can't stop talking to people. So please bear with me. That's probably not unusual for me anyway, though. Well, it's a personal pleasure and privilege to introduce the recipient of the 2023 SDB Lifetime Achievement Award. Each year, the SDB, the Society yearly confers this SDB Lifetime Achievement Award to a senior developmental biologist in recognition of outstanding and sustained contributions to science, exceptional mentorship, and outstanding service across a broad spectrum of areas. And you know, I was trying to think about how to reinforce what Anne has done, and so I spoke with several of the nominators and some of her trainees, and I asked what were the qualities and traits that she had, and I promised you I wouldn't embarrass you, but what kept coming up is passion, very passionate, very creative, analytical, determined to take on the tough problems and challenges, and caring. And that they stressed that that applied not just to her science, but to her roles in leadership, mentoring, training, and outreach. So I can't think of a more deserving person from that standpoint. So Anna's really been successful at every stage of her career. I think where I first really became aware of it is that she was a graduate student with Susumu Tonegawa at MIT, and she was studying immunoglobulin genes, and now we all know about enhancers and gene regulatory elements, but she identified one of the very first cell type specific enhancers and found motifs in it that we now know as the E-box. And then from there she did a short postdoc with Tom Maniatis, but she made the move. Ruth Lehman had just set up her lab at MIT, and Ruth said, could you imagine having one of your first trainees be Anne Ephrussi? And Anne joined, and she made the amazing discovery of Oscar, and through clever manipulation of its location, discovered that it was sufficient to induce germplasm. And I really encourage you to read, there's a wonderful article that the SDB put out of an interview by Anna Maria Carmo that really describes Anne's joy of the discovery, the eureka moment, which I think is wonderful. You know, so much of what we do doesn't work, but when it does work, it's just so special, right? And I thought that captured that moment. And that article also talks about her dreams, motivations, people who have inspired her. So it's really well written, and I really encourage you to look at it. But moving on from that work on Oscar, she really built her lab as a pioneer of understanding mechanisms of RNA localization and translational control. And over the years, her deep mechanistic insights have had significant impact, not just to the field of developmental biology, but cell biology and biochemistry. And she's widely recognized as really the intellectual and conceptual leader in these fields that are driving it forward. That's very important. But beyond that science, in addition, her accomplishments as a leader at the EMBL and a mentor have been really outstanding. She was a director and the dean of the graduate school. She also is committed to ethics and training, and so she teaches courses in that. But I think what everybody said was one of her most significant leadership contributions was the founding of the International Center for Advanced Training. And that organizes EMBL workshops, gives leadership training. She really built the principles of how that would work, that would develop career development opportunities for people. And that's part of her long-term vision of making sure that people are set up to be successful, are passionate about science, and could go on to whatever they do and carry science throughout multiple layers of our society. So you know, it's clear that Anne cares deeply about people. The testimonials are just remarkable from faculty, colleagues, senior colleagues at all levels. And I think that we all have a debt of gratitude to what she's done. So, Anne, congratulations on your award.
So I thank you, but I also would like to thank the Society for Developmental Biology for this tremendous award, to the people who have nominated me. This is a tremendous honor. It's extremely humbling. And I want to say right away that this recognition really doesn't go to me. It goes to all the people who have shaped me, trained me, with whom we've collaborated, my students. You know, it's a collective effort. And it's been a tremendous honor to be a scientist working in developmental biology in a mechanistic way, admittedly, to be paid by society to do what passions us. And this is something I tell my lab probably at least once a month. That doesn't seem like often, but I think they've heard it enough. This still really, it blows my mind. And I think this is why we owe, among other reasons, why we owe to society to do our utmost best to do science properly, with consideration, and really do it very carefully, economically, and remembering all the time how fortunate we are. So indeed, my talk today is in homage to all the persons who have really helped me and along the way, and friends. So first, I'd like to share a little bit about my personal trajectory as a scientist. It's not been without its bumps, I should say. As an undergraduate, I had the fortune to take a developmental biology course taught by Fotis Kafatos, who was a great teacher and a wonderful teacher, in whose lab I had the good fortune to work for my undergraduate thesis and more. And there I became definitively hooked on developmental biology, but also molecular biology and experimental research. And it was a really exciting time at that time. Recombinant DNA was just happening. This is the early 70s. There had been this moratorium where scientists had halted deliberately any work on recombinant DNA. This was a sign of responsibility. They really were concerned, as we are now, about what could be done with the tools that they were just developing. So this was in the midst. This was at Harvard. This was one of the places where this research was very active. Although I was planning to return to Europe, where I'd mostly grown up, for my graduate studies, I had a fellowship. I was going to Basel to work in a really nice lab. Then fate took a second bad hit on my family, and there was really my second parent died. You can read about the first in that little wonderful essay. And that's when I realized there was no reason to go back, and I stayed in the US. So after working for a year in laboratories and applying to graduate school in the US, which I hadn't done, I took a lot of courses in immunology and became fascinated by the question of self and non-self. And yet, being more molecular, when I learned about the immunoglobulin rearrangements that Susumu Tonegawa had discovered, actually also in Basel, I joined his lab at MIT as a graduate student there, initially to study recombination and try to understand how this somatic recombination happened in B cells. So now, soon after I arrived in the lab, and Susumu now was completely interested in T-cell receptor, which was not what I wanted to work on. And that, in fact, it made things tough. But at the same time, it gave me complete freedom to do what I wanted. And having a good network of friends and people to talk with, and hearing about enhancers, and indeed the first cellular enhancer was discovered in two labs, Susumu's and in Walter Schaffner's lab in Zurich. This was a fascinating question. An enhancer was a piece of DNA somewhere near a gene, and wherever you moved it, it would stimulate transcription of that gene. in a manner that was independent of position. And the immunoglobulin enhancer was super fascinating because it was cell type specific. This was completely new. The only enhancers to date that were known were polyoma SV40. They were viral enhancers. So here I was very lucky because I knew from my undergraduate days in the Harvard Biochem department, I knew George Church, who was a graduate student at the time with Wally Gilbert, a really incredible scientist and really a wonderful person too. And he had just developed a method that he called genomic sequencing. Sounds like he's been doing that all his life, but he called it genomic sequencing, and it made it possible to actually sequence total cellular RNA without any cloning by chemical modification of this blob of cellular RNA, restriction enzyme cutting, and then hybridization of these sequencing gels on membranes, so large sequencing gels, with incredibly hot probes. And that's why it says cooking with George. I actually was he was designing the probes. I was making at MIT the probes on my bench with 1 molecular shot. It was pretty crazy, but those were those days. And there, to our, I think, our amazement, because the idea here was now not to do this in the test tube, but do this in Petri dishes, and put the chemical modifier DMS on the cells, which would penetrate the cells, and modify the DNA in the cells. The DNA was still presumably bound by proteins. The cells were alive. And the idea was to footprint the immunoglobulin enhancer in B cells and in other cell types to see what this was. Because enhancers at that date, they were thought to be, or the cellular ones were thought to be, it was a chromatin effect. Some of you, old like me, may remember that chromatin was a dirty word. Anything you didn't understand, you would say, that's chromatin. Anyway, so, and I'm not joking. So remarkably, this experiment worked. And pretty soon, what we found was on one of these lovely gels, we identified these areas in the DNA ladder that were selectively protected from these chemical reactions that you see on the left. And these, we realized by looking at the sequence, had identical sequence, and indeed, they were most likely bound by proteins, which we showed by nuclear washes and whatnot. And we called these boxes 1, 2, 3, 4, elegant, and that's how they were called in the papers. We didn't call them E-boxes. And I'll just end by saying that, again, it's not always easy. After I defended, the tradition at MIT was that you'd give a lecture, a talk to the whole biology department. So, you know who was at MIT at that time. They were all in the front row. And at the end of my talk, one of the professors simply said, "I don't believe it," nice and loud. And I'd shown every possible control. I was a little rattled, but I simply asked him, "So what control should I do?" So it was tough. So it turned out that these were the sites later on that were found by others, Baltimore's lab, Sharp's lab, and others, to be the binding sites for basic helix, loop helix proteins. Now, for my postdoc then, I decided to study mechanisms of enhancer function in Tom Maniatis' lab, just back up the other way. But there I got quite distracted from transcription because Gary Struhl, a brilliant scientist, was a junior fellow at Harvard, and he was actually posted in Tom's lab. And he was working on morphogen gradients in the fly. This was brand new. The famous Heidelberg screen that you've already heard mentioned earlier today had just recently been carried out, in fact, at the EMBL by Eric Wischaus and Janine Slein-Volhard, saturating the Drosophila genome with EMS mutations to identify all of the zygotically expressed genes in the fly genome that were essential for embryonic patterning. And all those genes were now being sequenced in Tom's lab and elsewhere, and RNA and protein expression patterns of these segmentation genes, gap genes, were all being revealed. This was an incredible time. And so I was exposed to this and quickly lost interest in enhancers. But of course, it was clear from work going far back, and this was not my observation, but those people doing those screens, that actually, of course, the early events in embryonic patterning are already established before the zygotic genome is activated. That was shown in sea urchins by Driesch back in the late 1800s. Tennant, others had worked and shown that eggs are loaded with maternal so-called RNAs and proteins that really pilot the zygote through its first stages of development. And so obviously the next screens, and more difficult because these were F2 screens, were screens for maternal effect genes, where the genes that mattered were those in the mother and how she actually built the egg, and whether this, identifying the molecules in those eggs genetically that were responsible for early patterning. And at that point, I asked Tom if I could go to a conference in developmental biology. He kindly said, yes, let me go. And there I heard Ruth Lehman talk about the so-called posterior patterning genes. And she had been carrying out, as a graduate student in Yanni's lab, screens for, with the rest of the lab for these maternal genes. And this was one that was actually carried out by Yanni when she was still at EMBL. And Ruth spoke about these posterior group genes that selectively affected the posterior pattern, the abdominal pattern of the fly embryos. This was amazing, and I knew I had to change fields. I went back to Tom's lab directly from the conference, went to his office and told him, and he was so kind. And he said, you should do that. Call her immediately. So I made an appointment, and very soon thereafter left the lab and joins Ruth's. Ruth had just arrived, almost fresh out of graduate school, directly as a junior professor at MIT. And with all of these posterior group mutants, but one, Staufen, that Daniel St. Johnston was working on as a student in Yanni's lab, that affected abdominal patterning. And these were all genetic mutants, EMS mutants, whose general position on one of the chromosomes was known, but not much else. And I was very lucky. Others were working on Nanos and Pumilio, and I was entrusted with this little guy, Oscar, who failed to grow because of mutations in his mother. And in addition to lacking the abdomen, these embryos from Oscar females didn't have these posterior pole cells, which are the primordial germ cells of the fly. And this was really, really interesting to me. So I set out to clone Oscar. The general location, as I said, on the chromosome 3 had been mapped, but it was there, and I had to get going because there was no sequence genome. It was just mapped with deficiencies. And there was a question of carrying out in situ hybridizations to salivary gland chromosomes and in situ hybridization to identify, compare deficiencies with wild-type flies and try to see where the signal was lacking from cosmids. And finally, I did clone Oscar and found that it was localized as an RNA in the posterior of the oocyte and of the freshly laid egg, and where the pole plasm and germ cells form and the abdomen forms. And Oscar mutants, remarkably, failed to localize the RNA, and indeed, everything smelled right there. But then the real question was, if really Oscar was key to pole plasm formation, is it sufficient? It's clearly necessary, but is it sufficient? So to test this, we mislocalized Oscar to the anterior of the embryo by appending to the Oscar coding region the 3' untranslated region of bicoid RNA, which is targeted to the anterior. And lo and behold, 100% of the embryos developed this so-called bicaudal phenotype with a normal abdomen, because they still had the normal Oscar, but a completely mirror-symmetric abdomen in place of the head. But even more remarkably, transplantation experiments showed that these funny cells here, resembling those at the posterior, were actually capable of supporting germline development of a future zygote. So these were real germ cells. This was incredibly exciting. I just want to say here that the bicoid experiment I just described only recapitulated with modern tools a very classical experiment that had been done by Tony Mahowald and Carl Ilmensee much before, in which they took with a needle posterior pole cytoplasm, it's a clear region at the posterior of the freshly laid egg, and in fact unfertilized egg also, and transplanted it to an ectopic location. And they observed the formation of things that looked a little bit, had the features of pole cells, but they really didn't develop, and yes, and this experiment really sealed the case. So this was a very great tool also for mapping all of these posterior group genes and their functions relative to pole plasma formation and abdomen formation in germline. And so simply by crossing the oscarbicoid transgene into all of the posterior group mutants, it was possible to establish a hierarchy depending on whether they formed no abdomen, an abdomen only at the anterior, not at the posterior, would mean the gene is required for transport of the RNA to the posterior, or localization, or whatever the means were at the time. And thus we established a hierarchy with Oscar at the head of pole plasma assembly and accessory functions for localization of the RNA at the posterior. So when I moved to the EMBL in Heidelberg to start my own lab, my aim was to understand how Oscar is localized and translationally regulated such that its activity is restricted to the posterior pole, because what the oscarbicoid experiment also showed was it is extremely important to get that activity at the right place of the embryo, because otherwise it completely suppresses formation of the normal anterior structures. And so over the decades, we've been striving to understand how Oscar protein nucleates pole plasm, how the pole plasm instructs germ cell fate, and we've continued to work on these questions ever since. Now, Oscar is transcribed in the nurse cells of this germline syncytium surrounded by follicle cells. And upon splicing in the nucleus of the nurse cells, loading of the exon junction complex upon splicing and nuclear exports, the Oscar RNPs are transported by dynein into the oocyte by the dynein motor protein. In the oocyte, the RNPs switch to kinesin for their transport to the posterior pole, where the RNA is then translated. And I should say that the motors that were identified by Bill Saxton's group and Tom Hayes' group also working in the field. So it's thanks to them that we knew which motors were moving these different RNAs, and here Oscar. So once at the posterior pole, Oscar induces, the protein induces germline formation. We also have studied cell polarity and other aspects. But in recent years, our main focus has really been understanding how it has been focused on understanding how these RNPs are packaged into transport competent RNPs that are translationally repressed, and then how it comes about that upon localization, their translation is activated. And in the most recent times, our focus has also been on how the switch from dynein to kinesin is affected, how the motors associate, and how the switch is effected. So part of this is kind of boring, but it's identifying the signals, the cis-acting signals in the RNA responsible for these actions. And we identified years ago, Helena Jambor in the lab, a former PhD student, identified a stem loop structure in the Oscar 3' UTR that actually is responsible, we know now, for association of the unconventional RNA binding protein egalitarian. It's a double-stranded RNA binding protein that associates with bicaudal D, a component of the dynein-dynactin complex. And that is the signal that's responsible for transport from nurse cells into the oocyte. But then the question remained, what were the signals for kinesin, and then how does this switch happen? One of the first screens, we've of course genetic screens over the years in my lab, in a grandchildless screen for grandchildless mutants, so females that make embryos that don't have a germline specifically, we identified an interesting mutant in a tropomyosin gene, in which you could see that the dynein-dependent step worked perfectly fine. The RNA went into the oocyte, but the second kinesin-dependent step failed. We were able to later on build reporters to see the Oscar RNA moving here in blue. These little red with blue things moving are Oscar RNA particles on a polarized cytoskeleton here marked with EB1 cherry, which marks the plus ends. And in this way, we could also quantify the ratio of plus-ended to minus-ended movement. And there we found that in contrast to the wild type, where there's a plus-end bias of transport, so kinesin, there's a switch in the dominance of transport towards a minus end, which is dynein-based transport, explaining, presumably, why the RNA doesn't localize. And here I want to acknowledge the beautiful work of Daniel St. Johnston, who by live imaging, and without these reporters of polarity, also deduced the polarity of transport, and that tropomyosin also affected these steps. Now, we then found that through this assay that the association of kinesin with dynein is very dynamic. But even more interestingly, that actually in the absence of tropomyosin here on the right, there's a dramatic decrease in the percentage of kinesin molecules associated with Oscar RNA from approximately 30-some% to much less than half in the absence of tropomyosin. And this indicated that somehow tropomyosin is involved in the association of kinesin with the RNA. We further discovered that tropomyosin is an atypical RNA binding protein. It's an unusual tropomyosin isoform, and that it binds the Oscar 3' untranslated region, and also that this tropomyosin binds to kinesin, both in vitro and in vivo. So here with the RNA, kinesin, and tropomyosin seem to be the linker. So this was all fine and good. We now seem to have what we needed to transport the RNA. The problem was that we found by imaging that actually the kinesin already associates with the RNA in the nurse cells. So this posed a conundrum. How can dynein function if the kinesin is also associated? And then in the oocyte, once in the oocyte, how is it that dynein is no longer affecting the transport to the posterior pole? So I'll skip a little bit fast through these slides just to say that kinesin molecules, when they're inactive, actually are auto-inhibited by folding back of the regulatory tail domain onto the motor domain and precluding the binding of the motor to the microtubules. And this inhibited state is typically reinforced by the binding of a kinesin light chain, which is also usually the cargo binding domain, which further locks the inhibited state of kinesin. Now, the problem is that it was shown by Daniel St. Johnston's lab, Isabel Palacios, that in fact the kinesin light chain in the fly is not involved in any of these germline events in transport of Oscar, or in fact others. So we wondered then, well, could tropomyosin be acting as an alternative light chain? And I'll fast forward just to say that we identified the region of interaction between the proteins. We crystallized the coiled domain of the tropomyosin, found that it forms dimers, and then obtained the structure of kinesin in complex with tropomyosin, which we found forms a tripartite structure consisting of 2 parallel kinesin chains and 1 antiparallel tropomyosin. And if one looks at the electrostatic projection of this interaction region, one sees it is highly basic, positively charged, suggesting it might interact with RNA. And more remarkably yet, both tropomyosin and kinesin showed a low affinity for RNA in the high nanomolar range. That's not particularly high, but when they are together, actually the affinity rises significantly. So it seems that when these two proteins that one doesn't know as RNA binding proteins assemble, they actually bind RNA. So then to test the ability of kinesin and tropomyosin, the complex, to transport the RNA, and to understand more about this regulation, a postdoc in my lab, Simona, established in vitro motility assays using fluorescently labeled recombinant kinesin and full-length tropomyosin, kinesin, and in vitro transcribed Oscar RNA, which you can see here moving on immobilized microtubules on slides. So this is completely reconstituted in vitro. And for this work, we collaborated with Simon Bullock and Mark McClintock at the LMB in Cambridge, because Simon, who is just an extraordinary scientist working in the same field, initially a student of Rosa Beddington's, by the way, who, when she passed away, moved to David Ishhortz's lab and started working on RNA transport. He had actually fully reconstituted with Mark McClintock the dynein-based transport of other RNAs, apically localized RNAs in embryos. So, and in fact, Simona is a jointly supervised postdoc working in my lab also with Simon. Simona also performed biochemistry and biophysical measurements, and we were aided with the small angle X-ray scattering and NMR by Janos Hennig at the EMBL and Bernd Simon, who's actually just moved to Yale. And by performing these reconstitution assays with full-length kinesin, tropomyosin, and this RNA, we were able to find that in vitro, the kinesin can associate with Oscar RNA and be motile. The enhancement of the binding with tropomyosin was not seen in this assay, but what was very clear, if we looked at the velocity and the properties of the motility, if you look in green here, or blue, turquoise, in the presence of tropomyosin, the runs of kinesin with Oscar RNA were quite rapid, 0.2 microns per second up to almost 0.8 microns per second, which is incredibly fast for a reconstitution assay. But in the absence of tropomyosin here in purple, the velocity dropped by a factor of greater than 3, down to 0.68 microns, 68 microns per second. So clearly, somehow the tropomyosin has an inhibitory effect on the kinesin. So this was interesting, but knowing that tropomyosin is a cargo adapter, isn't it a bit counterintuitive that it also inhibits kinesin? So as I mentioned previously, kinesin and tropomyosin are loaded already in the nurse cells onto Oscar RNA when the dynein-based transport is required. So we reasoned that during this process, the binding of tropomyosin to kinesin might inhibit the kinesin and promote the dynein-based transport, by preventing a tug-of-war between the two motors. So finally, to test if this was the case, Simone and Mark adapted the assay such that it would work, the conditions were sufficiently okay for the two motors, which isn't, they don't like the same thing in vitro, and carried out the assay with RNA, dynein, kinesin, and in the absence or presence of tropomyosin. And what they found was that actually in the absence of tropomyosin, the kinesin movement, motile events with Oscar were very few, and very few went in the posterior, in the plus-ended direction. The majority were actually minus-ended with dynein. However, upon addition of the tropomyosin, the motility vastly increased, particularly in the minus-ended direction, so that kinesin was associated with the RNPs, but was actually not moving in the plus-ended direction. It was held off by tropomyosin, and was then, the RNPs could then translocate in the minus-ended direction, as you can see here. So, in vivo, this mechanism that we've been studying would allow the minus-end directed dynein-mediated transport of Oscar from the nurse cells into the oocyte to proceed without a tug-of-war with kinesin, while positioning that motor already in an inhibited state on the RNA, such that when it is transported into the oocyte, it is positioned to drive the RNP transport now towards the posterior pole during mid-oogenesis. Other questions have been then, do you stop dynein transport in the oocyte? Here we have found that actually the Staufen RNA protein displaces the egalitarian, and this contributes to inhibiting the dynein-based transport once the RNA is in the oocyte. The outstanding question, which Simone will continue to work on in her own lab, is then how does one disinhibit the kinesin? How does one alleviate the tropomyosin-based inhibition of kinesin? And that really remains to be understood. So now I will actually escape and go directly to my acknowledgement slide because I have far too many. I was going to tell another story that I'm very excited about, but I think that I will just go here to the acknowledgements. And just say that we have also found, many people have been studying the question of condensates and phase separation. This was a field that certainly I wasn't too excited about, and thought it was really overblown. And I thought, well, let's see if it really matters what state the RNPs are in. We decided to explore this and found actually that the small, which are very small limited Oscar RNPs in the oocyte are actually completely spherical, which means that they have phase separated. This is the only way, and that they've been liquid. They have been liquids because this is how you adopt a spherical shape is by minimizing volume. But they were completely solid. And what Boza in the lab, a brilliant postdoc, did was he managed to liquefy these granules genetically by attaching a low complexity domain that induces liquidity, and he found that this completely disrupts Oscar regulation. So it does matter, the material state of RNPs does matter. Now I'd like to finally end by thanking my current collaborators in my lab. My lab is very, very small because unfortunately, due to mandatory retirement in Europe, my lab is shutting later this year. My students, postdocs will continue, and all our colleagues in the field, and I will enjoy reading about their work, hopefully discussing it as well. But this is my little group. The work I told about was that of Simone Heber, who is here. Also Mojnak, I alluded to others, and I want to thank my colleagues whom I've mentioned during my talk, our core facilities. All my fellow travelers, these are all my PhD students and postdocs, not all the trainees, undergraduates, and so on. And these are my students, postdocs, and technicians who have worked with me in 31 years. Many people say, oh, that's not many. It's only about 63. I'm actually well, it's over 30-some years. I believe in having a small lab, especially when everyone works on a different project. The only way to be able to learn about these things, think about them, and to contribute is to really be able to keep the lab small, and this encourages collaboration, which is dynamic. Things change depending on what we find, and it is so enriching to work with others. And I would like to thank you for your attention, and again for this award.